Estamos ao vivo.
Good afternoon, everybody. It's a really pleasure for us to receive today for our seminar, Dr. Maria Caetano and Dr. Guillermo Docena, in this seminar supported by ALACI. And today we'll talk about mucosal immunology. So our first speaker is Maria Caetano. She holds a medical degree from the Federal University of Minas Gerais, a master's degree in microbiology from the Federal University of Minas Gerais, a PhD in immunology from the University of São Paulo, and postdoctoral fellowship at Harvard Medical School in Boston. She was visiting researcher at Universidad de Bologna in Italy, at Rockefeller University in New York in 2016. She was a member of the board of the Brazilian Society of Immunology from 2011 to 2013, but presently she is the president of the Brazilian Society of Immunology. In addition, she was also member of the advisory board of biological science and biotechnology of APNID, the advisory committee of immunology of CNPq, and is the coordinator of the Subcommittee of Mucosal Immunology Nomenclature of IEYS since 2019. She is currently a full professor of immunology at the Federal University of Minas Gerais. She has experience in
I think there is a problem with Claudia's internet.
Are you listening?
Yeah, now
Yes,
we
yes.
are. Yeah.
Okay. So I have already introduced you, but I can introduce again. So have you heard?
No.
Okay.
Yeah, because you were at the end. That's okay.
So,
I'm more than introduced.
Okay. So have you listened that I talked about you, no, Anna?
Yes, yes.
Okay. So it's a pleasure for us now to listen to you, and we are ready to listen.
Okay, thank you very much, Cláudia, and thanks to the organizers, to Valderez and the ALACI for the invitation to participate in this series of talks. I'm going to share my screen. Hope you're seeing this.
Yes, Anna.
Yeah, it's okay. Okay, so today I'm going to talk to you about the connection between the gut mucosa and the type 1 diabetes. And this slide is just to introduce the subject and to show you why the intestinal mucosa is so important for the body. Well, we know that the intestinal mucosa is a large surface, it's 300 square meters, so it's the largest surface of contact with of the body with the external world. And it has, let me pick up if I can find I'm going to present with the arrow. Okay, it's a very large surface. It's the largest one actually, and there is only a single cell layer that separates the external world, in this case the lumen, from the body, which is the epithelial barrier of the intestine. And in addition to that, there is a large collection of lymphoid cells and immune cells that inhabit this organ. And here you can see there are lots of immune cells, lymphocytes, macrophages, dendritic cells, eosinophils, mast cells that are here in the villi, in a compartment we call lamina propria, and also the intestine has these structures which are similar to lymph nodes, that are the pyopathes in the small intestine that are also organized lymphoid tissue. So actually the intestine is the largest lymphoid organ in the body. And what is also very remarkable about the intestine is that this large surface with this huge amount of lymphocytes and immune cells is constantly challenged by two kinds of what we call natural antigens. One is represented by the dietary proteins, and the other one are the gut microbiota. Just to give you an idea, we are colonized by 100 trillion bacteria that we host in our gut, in our intestine. And also we ingest more or less 190 grams of proteins a day. And with this large load of antigen stimulating all these cells in the gut intestine, you would imagine that you have a huge reaction, but actually the immune response that is common in the intestine induced by these antigens is not an inflammatory one, is rather an anti-inflammatory response that is called oral tolerance. Oral tolerance is a phenomenon that has been described like more than 100 years ago. And this means that we are tolerant to the antigens that we eat every day and also to our intestinal microbiota. Oral tolerance can be like experimentally reproduced by a protocol like this one I'm showing here. So here, in the control group, if we immunize a mouse, for example, with ovalbumin, an antigen, a protein, this is a dietary protein, with adjuvant, what we get is a classical antibody response and a cellular immune response. But if you feed these mice with ovalbumin, with the same antigen before we immunize, then what we have is an inhibition of this inflammatory immune response. And this is what is called oral tolerance. So it means that this response can be measured like in the serum or at distant sites. So oral tolerance is a response that is induced in the gut, but has systemic effects. And we know now that oral tolerance is induced mostly by cells that have a regulatory role that control inflammation called regulatory T cells. These regulatory T cells are mainly the CD4 positive T cells. Most of them are FOXP3 T cells that are induced in the gut. But there are some that are also positive, have TGF-beta in their surface, and they are called LAP-positive cells. These cells mediate their anti-inflammatory inhibitory effect mostly by the production of anti-inflammatory cytokines such as IL-10, IL-35, TGF-beta. And the cytokines and the cells are able to inhibit effector inflammatory responses driven by Th17, Th2, macrophages, which are cells that are very, very abundant in the intestine. So regulatory T cells and the tolerogenic response in the gut is responsible to keep a balance between inflammatory and anti-inflammatory mechanisms and to keep gut homeostasis. So to keep a silent and balanced response in the gut. And not only that, but we know that this activity, this whole activity that goes on in the intestinal mucosa has an impact in several distant organs and tissues in the body. One that we are going to talk today that is very important is the pancreas. And there are already descriptions showing that the gut microbiota and also the intestinal permeability have an impact and are involved in the development of type 1 diabetes, which is a disease that affects the beta cells in the pancreas. These descriptions are related to usually describe the influence of gut microbiota and some other antigens in the gut when the disease is already established. So we know that gut microbiota, for example, is very important for the pace of the disease, for the progression of type 1 diabetes. And but we don't know exactly how these antigens are important for the onset, for the triggering of the disease. Type 1 diabetes, as I told you, actually is not an uncommon disease. It represents 10% of all the diabetes cases, and diabetes is prevalence is very high worldwide, and it can start in kids. It can start even in babies. So it's a disease, it's an autoimmune disease that the incidence has increased a lot in past years, and especially in children. The physiopathology, I mean, how the disease develops, it's, as I told you, is an autoimmune disease. So it's triggered and it's maintained by autoreactive lymphocytes, especially T cells, CD4 T cells, autoreactive CD4 positive and CD8 T cells that that react with antigens in the islets, in the Langerhans islets, so in the beta cell, the beta cells in the islets, in the pancreas, and it starts with an inflammation. So these cells start mediating an inflammatory reaction in the pancreas that we call insulitis, and it ends up destroying these beta cells. We know that autoantibodies can have a participation in the inflammatory reaction, in the destruction of these beta cells, and ultimately we have an inability of this organ of the pancreas to produce insulin, because beta cells are the ones that are responsible for the production of this hormone. This will lead to a hyperglycemia, and hyperglycemia would have a huge impact in several tissues and organs, and this is part of the pathogenesis and of the development of the disease. So the disease goes on with retinopathy, poor blood flow that affects nerve and kidney, and with kidney skin alterations, and it's a very chronic disease that can start, as I said, very early, and it can go for decades. We know that type 1 diabetes, this is valid for all autoimmune diseases, have a genetic component, and also environmental factors are important for the development of the disease. We know that there are genes that are responsible for susceptibility. It's called Iddm genes. The most important ones are the MHC genes. So HLA-DQ is the most important genes that confer susceptibility to diabetes, and there are also protective genes that confer protection, such as HLA-DR3. Other genes that are responsible for immune regulation are also involved in the disease. But in addition to this genetic background, we also know that there are environmental factors that play an important role in the development of the disease, because studies with identical twins show that one of them usually develop the disease before the other. So it's clear that environmental factors play a role. And of course, food components and microbiota antigens are like good candidates as environmental factors in the development of type 1 diabetes. And that's what we are going to talk today. The pancreas, which is the target organ for the destruction that starts and is responsible for type 1 diabetes, is a very interesting organ and has a very close relationship with the gastrointestinal tract. Although it's not part of the TGI, it sits there in the abdomen in a very close proximity to the mesentery, to the duodenum, contacts the spleen, the stomach. It participates in the digestive process because it secretes exocrine enzymes like that are present in the pancreatic juice. And it's also involved in metabolism because it's an endocrine organ and produces, for example, insulin, which is the important hormone that is insufficient in type 1 diabetes. Also, the pancreas has a common embryonic origin with the duodenum in the small intestine. Not only that, but we know that the so-called pancreatic lymph node drains at the same time the pancreas and the duodenum. So duodenum, this region here in the intestine, and the pancreas share the same lymphatic drainage. And it's interesting because these lymph nodes, they are separated from the chain of mesenteric lymph nodes that drain the other parts of the small intestine. Not only it drains the pancreatic lymph node drains the duodenum, but we know now that the duodenum, that the lymph nodes, the duodenal lymph nodes are very different from the other ones from that belong to the mesenteric chains in terms of activity as well. We know that when you examine the T cells and the dendritic cells and the immunological activity that goes on in these lymph nodes, most of the regulatory T cells are produced in the duodenal lymph nodes, the ones that also drain the pancreas. And if you look at the frequency of regulatory T cells in these lymph nodes and compare with the other ones, they're much higher. And at the same time, you have a lower frequency of inflammatory T cells like Th17 cells there when compared to the other ones. So there is a compartmentalization in the immune response in the gut that is reflected in the lymph nodes that drain this region, and the duodenal lymph nodes are the ones that are responsible for the generation of regulatory T cells for dietary antigens and antigens that are in the gut, and for the oral tolerance induction as a consequence. So our question for this work and this project that was carried on by two PhD students in my lab was how immunological events taking place in the intestinal mucosa would interfere with the onset of type 1 diabetes. Why? Because as we see, there is a close correlation between pancreas and the intestine. And of course, this would mean something about the type 1 diabetes. That's what we wanted to investigate. For that, we used a mouse model that is very interesting to study type 1 diabetes, which is the NOD mice, the non-obese diabetic mice, because these mice develop spontaneously the disease. You don't have to induce it. And also, they have a lot of Iddm genes, as in humans, and the MHC class 2 of these mice is an ortholog of HLA-DQ that is associated with susceptibility in humans. So disease in these mice usually starts at 12 weeks. So you have a period when the mice has no signs of insulitis or hyperglycemia whatsoever. At 12 weeks, they start having the insulitis, let's say, so the inflammation of the islets in the pancreas. And at more or less 16 to 20 weeks of age, they have destruction. You can see destruction of these islets, and then hyperglycemia, and then diabetes. As a control, we compare with NOR mice, which is a mouse that has the same genetic background of NOD mice, including the same MHC, but has some resistant genes, so the mice does not develop diabetes. So the experimental design is this. We compare NOR and NOD mice at an age that is very early, so 4 to 6 weeks, right after weaning, with mice that are at the time when they develop diabetes, which is 16 to 20 weeks. And we measure hyperglycemia and consider diabetic mice when they have a glycemia above 250 milligrams per ml. The first thing that we saw that we follow was, of course, the development of diabetes in these mice. And as you can see here, NOD mice start developing diabetes at 12 weeks of age, and then the incidence increases as they age. NOR mice did not develop diabetes at all. And then you can see by the histology here that at 4 to 6 weeks, you have no signs of inflammation in the pancreas of NOR or NOD mice. And then in NOD mice, at the pre-diabetic mice, you have insulitis with the inflammatory infiltration here. And the diabetic mice is the one that has a huge inflammation and destruction of the beta islets, the beta cells. So the question was how this development of diabetes is related to if it is related to some alterations in the intestine. So we decided to look in the intestinal mucosa in these mice and see whether they develop some alterations that could be related to the development of diabetes. And to our surprise, we saw the first thing that we looked for was the mucosal barrier. And the mucosal barrier, when we talk about that, is epithelial cells and cells that produce important components of the barrier, which in this case, we are looking at the goblet cells that produce the mucus. And for our surprise, we saw that very early, like in mice to 4 to 6 weeks of age, so after weaning, much before they have any signs of insulitis or diabetes, they already have a reduction in the frequency of goblet cells, in the frequency of MUC2, which is the main component of mucus, and also in the amount of in the concentration of mucus in the gut, in the intestinal, especially in the small intestine. And a master student in the lab, Carla Catarina, just looked at mice much before that. She looked at actually at neonatal mice at 1 week of age. And in these mice, we already see a very striking reduction in the expression of proteins in molecules that are responsible for the connection between epithelial cells to compose the epithelial barrier. So this suggests that there is a very early defect in gut permeability in these mice. So as you can see here, all these molecules are already altered and are decreased. The expression is decreased in NOD mice when compared to NOR mice at 1 week of age. So we decided to look at other components of the mucosal barrier, and one of them is the IgA, the secretory IgA, that is present in the mucus and is very important to not only to keep the pathogens away from the body, but also for the composition of the microbiota. And as you can see, at 4 weeks of age, 4 to 6 weeks, we already have a huge reduction in the production of secretory IgA in NOD mice when compared to NOR mice. And of course, in mice that are older than that, you also have this defect. Not only the production of secretory IgA is defective, but when we look at the microbiota, the bacteria in the microbiota, and the binding of this IgA, so the frequency of bacteria that is bound to IgA, as I said, IgA is very important to keep an homeostatic composition of the microbiota because it binds to the microbiota. We see that there is also a reduction in that. So this is already a clue to say that these mice probably have a defect in the composition of their microbiota as well. And this is very early as well, and it happens in the small intestine. And then we decided to look at whether we could find inflammatory cytokines of a kind of inflammatory profile in the intestine at that age. And here we can see that we have an increase in some inflammatory cytokines, such as IL-6 and IL-12, but later on in pre-diabetic mice in the gut. This is a measurement of expression of the cytokines in the gut mucosa. And, but for IL-10, for example, which is a very important regulatory cytokine in the intestine, we have a reduction, a significant reduction in NOD mice as compared to NOR mice in very early, 4 to 6 weeks of age. And this could be reproduced when we measure the protein, the IL-10 by ELISA as well. So we have a defect as well in the production of cytokines and a more pro-inflammatory profile very early in these animals. The next step was to look in the gut microbiota. We did that with the help of Daniela Carlos from USP, Ribeirão Preto. And the first thing we saw is that when compared NOR and NOD mice at 4 weeks of age and the diabetic mice, is that we have a translocation of microbiota in NOD mice when compared to NOR mice. And this translocation was measured in the pancreas. So we have bacteria that should be in the intestine, in the intestinal microbiota that was found in the pancreas. And most of all, what we saw was we have a very striking difference in the microbiota when we compare NOR and NOD mice. Of course, this was already reported for mice with diabetes, and that's what we see here. This is pre-diabetic NOD mice, and we can see that there are some alterations. We have an increase in the abundance of more bacteria with more pro-inflammatory profile when compared with NOR mice. But what we found is that those alterations in the gut microbiota, they start very early, at 4 weeks of age, when the mice is weaned, and is the time when they establish their permanent microbiota. We already have dysbiosis. We already have an increased abundance of a more pro-inflammatory profile of bacteria, in a decrease in the anti-inflammatory and producers of metabolites that have anti-inflammatory effects, such as short-chain fatty acids. So the more pro-inflammatory profile starts very early in these mice. Then we decided to do an experiment to see what was the role of to confirm whether the microbiota would have a role in the alterations that we saw in the gut, especially in the gut barrier function. And then we did an experiment, which is a cross-fostering experiment. We took the NOD pups and put them to be breastfed by NOR mice. So these animals here, the white ones, they were born from NOD mice, but they were breastfed from by NOR mothers. And then we look at them 24, and they were transferred when they were 20 hours old, so was exactly in the same day. And 4 weeks later, when they were weaned, we look at the alterations in the gut mucosa to see whether they were reversed by the cross-feeding. And what we can see here, we use the mucus production as a readout. And as you can see in the small intestine and in the colon, we have a reduction in the mucus production goblet cells number in NOD mice, as we already have shown, in the small intestine and colon, but mouse, the pups that were breastfed by NOR mice, they recover and they are similar to their mothers, showing that two things can happen here. Either the microbiota of the mother is installed and interfere with the development of the gut barrier function, or some element in the milk itself is playing a role. And we also look, of course, in the dendritic cells and regulatory cells in these mice, and then we took the duodenal lymph nodes, the ones that drain both pancreatic and duodenal, and look at the frequency of regulatory T cells and dendritic cells that induce regulatory T cells, tolerogenic dendritic cells. And what we saw was in the duodenum, we have a reduction of these tolerogenic dendritic cells in NOD mice, in pre-diabetic mice, and we also have a reduction of regulatory T cells in the lymph nodes that drain the duodenum in pre-diabetic and diabetic mice. So it seems that you also have a defect, but later on, in the regulatory function of T cells in these mice. And of course, we look at oral tolerance induction in them, and we look at 4 weeks of age to see how early it would happen. And here is an experiment in NOR mice that we have. Here's the immune mice, the one that we induce tolerance to ovalbumin. So we feed ovalbumin and immunize with ovalbumin, and we see that we have a classical oral tolerance response, a reduction in the response, and this didn't happen in NOD mice at 4 weeks of age. So they have a defect in oral tolerance induction. And this is true also. Natalia Pinheiro Rosa, a PhD student in my lab, also showed that they also have a breakdown of oral tolerance to the gut microbiota. So what we have shown was that there are defects that are present in the gut mucosa of these mice that are related to the gut barrier and also to the regulatory T cells and neurotolerance induction and regulatory mechanisms that precede the onset of type 1 diabetes. And this is really important because it could represent a way to manipulate or to intervene in susceptible children, for example, and to create a kind of therapeutic tool very early in age. Just to acknowledge the students that participate in the students, our collaborators, and this is a very old picture of the lab, and this is just to show that the first meeting of LAMIG, the Latin American Mucosal Immunology Group, Guillermo is here and Cláudia is here, happened in Belo Horizonte in 2016, and those are the invited speakers, and was the first meeting of LAMIG. It was the second workshop on mucosal immunology. So thank you very much.
Thank you very much, and a beautiful talk, very, very nice. So I will introduce Guillermo, and after Guillermo, we can open for the questions, okay?
Okay.
So Guillermo Docena is a senior researcher at the National Council for Scientific and Technical Research, graduated in biochemistry faculty of exact science from the National University of La Plata, and holds a PhD from the same institution. In 2005-2007, he did postdoctoral internships at the Institute of Cell and Molecular Science in Queen University, London, England, under the supervision of Dr. Thomas MacDonald, in studies related to the immune response and inflammatory gastrointestinal diseases. Since 2007, he leads the immunopathology and immunointervention in intestinal inflammation research group. He is currently assistant professor of the area of immunology and microbiology in charge of immunology subjects taught for the careers of biochemistry, pharmacy, and biotechnology and molecular biology of the Department of Biological Science. He is also permanent secretary of the Latin American Association of Immunology, a member of the Education Committee of the International Union of Immunology Societies, director of FOSIS, Federation of Clinical Immunology Societies, Center of Excellence, president and of the Food Allergens Platform, coordinator of the Food Safety Network, Food Allergens Group, and has been president of the Argentina Society of Immunology. Guillermo, thank you very much for accepting our invitation, and please, we are ready to hear you. Thank you.
Okay, Claudia, thank you very much for your kind and friendly introduction. It's always a pleasure for me to share this type of activities and any other activities with my Brazilian friends. And I would also like to thank ALACI and SBI for this invitation. So I will share my screen, and I will show you today some stuff we have obtained the last year. Some of them have been already published, but other results were recently obtained, even though the pandemic. Let me see. So I will talk about human stuff after Anna's presentation, and we are trying to get information about the synthesis of IgE in the gut of food allergic patients. And of course, this is the results that I will show you today are the first to be published in terms of human stuff. And of course, we have several questions and some other certainties. So you know that the mucosa is defined as an organ that is in contact with the environment. And so it means that it is continuously and some of them intensively exposed to a myriad of antigens. And regarding the digestive tract, the gut is the main organ, and as Anna said before, it had a huge surface area with almost 300 square meters, and most of this area is concentrated or is located in the jejunum and ileum. And today I will talk about some findings that we have in the colon. So I guess you know that food allergy is an abnormal immunological response to food proteins, and it should be related with any clinical manifestation. Otherwise, it's only allergic sensitization. But the point is that in the last decades, it became a worldwide health problem because its incidence has increased. And today, the IgE-mediated food allergy, which are the most severe reactions, may affect millions of people, mainly in the pediatric population, but nowadays also in the adult population. And since the 19 and 2000, we have witnessed an increase in the allergies incidence with epidemic impact until or after 2010. We are in the second epidemic wave of food allergies in different regions of the world, but not only changed the prevalence, but also the presentation and the severity. And today we have more food-related anaphylaxis than what it has been seen decades ago. And as judged by the high efficacy of the anti-IgE treatments in allergic patients, we can say that the IgE is an appropriate target for treatments, but there is a black box in all this story because we don't know anything, or we have very few information about the origin of IgE and mainly the memory IgE in humans. And that is because the access to the tissue where IgE is produced is very scarce, very infrequent because of ethics, and the cells that are bearing IgE in periphery are very low in frequency. And so you know perhaps that the results obtained by Maria Curoto and Juan Lafay in the States, and most of the information we have about memory IgE is coming from mice studies. And as you know, allergy is not spontaneously produced in mice as it is in humans, so perhaps the origin and the biology of IgE is quite different. Regarding human stuff, we only have some papers, very few of them, in which it has been demonstrated that IgE is produced in bone marrow, in adenoids, nasal mucosa, and bronchi, but nothing has been published in terms of production of IgE in the human gut. This is a paper we published last year, and here we have demonstrated that IgE is produced in the human gut. This is a work in progress, so I will show you some results about that. And this is the main goal of our project, to characterize the stroma of polyps. I will show you why I'm talking about polyps. There is a cell infiltration, as I will show you. IgE is produced within these polyps, and this may have an impact in different therapeutic strategies to modulate IgE synthesis in the gut. So this work has been mainly done by Karina Canciani. She got her PhD last year. We studied more than 100 patients who were selected at the Child Hospital in La Plata. Patients had rectal bleeding and suspicious of milk allergy. This was the age of the patients, more than 2 years and less than 6. We analyzed the personal and family medical history related with allergy and atopy. Skin test and IgE were assessed in all patients, and colonoscopy was indicated in all patients to rule out any other cause of rectal bleeding. And what we found was that almost in 50% of the patients, they have polyps, mainly located in the rectum. Most of them have a single polyp, pedunculated of different size. And we analyze the nature of these polyps. Some of the sample, or half of the sample, were sent to the histology units at the hospital. They analyzed different tumoral markers and they are negative for these markers, so it's not a tumoral polyps. And then we receive another part to perform all the experiments I will show you now. And in addition, we could get a biopsy of the surrounding tissue, which is our internal control. And all the patients underwent a restriction diet just to confirm or to discard the possibility of food allergy. So I will not go into detail. This is already published in the allergy paper, but I can say that in most of the patients, 90% of them, we could detect food-specific IgE in serum. Almost 80% of patients had total level of IgE elevated compared to the normal value. Skin test was only positive in 1 patient. Very few patients show elevated peripheral counts of eosinophils, and most of the clinical history was related with asthma, dermatitis, urticaria, rhinitis, and milk allergy. So when we analyze the tissue in the polyps and in the surrounding tissue, we could observe that within the polyp, from now PT, so this is the tissue in the poly, we could see a huge or a dense cell infiltration that was not observed in the surrounding or in the adjacent mucosa of the same patients, and also compare with normal patients or healthy patients. And between all these cells, we could detect a huge amount of mononuclear cells, but also eosinophils and mast cells. And we confirm the nature of these cells by a differential staining and also here by confocal microscopy. And so we could count the eosinophils, and as you can see here, there is a significant increase in the eosinophil count only within the polyps and not in the surrounding tissue, which is quite striking. But then the polyps reveal a huge fibrosis, edema, and also we could detect that the content of IgE within the polyp is significantly higher than in the adjacent tissue. So we can see that within the polyp there is an inflammatory infiltration with eosinophilia and fibrosis, which are hallmarks of allergic inflammation. To address this point, we assess why eosinophils are being recruited to the tissue, and for that we analyze the expression and the production of different chemokines, and here I can show you 2 of them, Rantes and eotaxin 3, and as you can see here, Rantes is not differentially produced within the polyp and in the adjacent tissue, while CCL26, which is the main attractor for eosinophils, or eotaxin 3, is mainly produced within the polyps. This is in terms of transcript level and also at the protein level, and there is a significant increase of this chemokine within the polyp, and by fluorescence or confocal microscopy, we could see that CCL26 is mainly produced in the epithelial compartment. So from these findings, we can say, or we could see that the main eosinophil attractant is produced by epithelial cells only within the polyp. And trying to go further to understand why is it produced, we analyze different cytokines also in the polyp and in the biopsy at the protein level and also at the expression level, and we could see an increase in this transcript coding for IL-4 and IL-13, and not for IL-5, which was quite or rather striking. gamma interferon remain unchanged when we compare the content in the polyp and in the biopsy, but nevertheless, the ratio IL-13 gamma interferon was increased within the polyp. We obtained the same results in terms of type 2 cytokines at the protein level, with a high production of IL-4, IL-13, and a high ratio of IL-13 and gamma interferon. So we can say that the polyp tissue is dominated by type 2 cytokines. We also analyze some other cytokines, and here I can show you two of the alarmins, IL-33 and TSLP, which are closely related with the early steps of allergy induction. And we could see here that IL-33 is also mainly produced within the polyp, mainly in the epithelial compartment. And we can see here that TSLP is also produced by the intestinal or colonic epithelial cells. ST2, which is the receptor for IL-33, is also more expressed in the polyp than in the surrounding tissue. And what we have found is that fibroblasts, which I told you before, there is a dense infiltration or a dense presence of fibroblasts. They are producing IL-33, as you can see here in the nucleus. And so we can say that IL-33 and TSLP, which are drivers of the allergic reaction, are also increased only within the polyp. And perhaps the epithelial compartment is involved in this production. So trying to understand what is the role for the cytokines and the eosinophil recruitment, we expose CACO2 cells, which are colonic human epithelial cells, to different pro-inflammatory stimuli, like here the flagellin, IL-13, and gamma interferon, and also IL-5. And we could see that CACO2 cells in the presence of IL-13 is inducing the expression of CCL25 and also the secretion of CCL25, and try to confirm this finding, and that is produced through the IL-13 receptor. We incubated the cells with IL-13 and also with dupilumab, who is a biological or monoclonal antibody specific for the IL-4 alpha receptor, which is part of the IL-4 and IL-13 receptor. As you can see here, there is a significant suppression of CCL26 production. IL-5 is not inducing CCL26, and so eotaxin 3 is induced by IL-13. And then we perform a kinase array, trying to find any kinase that could be involved in this mechanism. And we could see that STAT3 is induced by IL-13, also by gamma interferon, and then we confirmed this finding by immunoblotting, and so we incubated CACO2 cells with IL-13 and gamma interferon, and we could see that STAT3 is phosphorylated with IL-13, and also STAT6 is phosphorylated, and this is very well described in the literature. So, trying to understand what is the intracellular pathway involved in the induction of the secretion of CCL26, we incubated the cells with ruxolitinib, which is a JAK1 and JAK2 inhibitor. And as you can see here, there is a dose-response inhibition of the secretion of CCL26. So we can say that STAT3 and STAT6, which undergo through this YAC kinase pathway, are involved in the secretion of eotaxin 3, and this is induced by IL-13, which was previously demonstrated that it's upregulated in the polyp tissue. So this I will go very fast. This is this part of the work was done by Cecilia Muglia. She has characterized T cells within the polyps. We could see that T cells are specific for milk proteins. We isolated, or we obtain milk-specific cell lines and also the clones, and we could see that they respond to the milk allergens. They produce IL-13, they do not produce gamma interferon. The same for the cell lines and for the clones. All of these cells are TCR alpha beta positive. And this is a quite striking result that we obtain, that all of these cells, or most of these cells, they are expressing CCR9 chemokine, which you know that it is a chemokine that it's expressed by the cells that are being recruited to the small bowel. I didn't tell you before, but we obtain cell lines and clones also from the peripheral blood of the same patients. And in all cases, CCR9 are increased in these cells, which means that perhaps the circulating cells will migrate to the small bowel or also to the colon, because we could see that there is a significant increase in the cognate chemokine within the polyps, CCL25. We observe significant induction of this chemokine only within the polyp and not in the biopsy. And this is quite interesting because we don't know if the cells are being attracted to the polyp from the periphery. Perhaps they are being generated in small bowel, or perhaps they are being generated here in the colon within the polyps, and then they migrate to the other tissue or to the other mucosa. So the last part of my talk, I will talk about IgE. As I told you before, the levels of total IgE and milk-specific IgE are increased only within the polyps and not in the surrounding tissue. You can see here the IgE positive cells. As you can see here, they are not observed in the adjacent mucosa. And we could see that there is a significant high frequency of these IgE positive cells only within the polyp. There was a correlation between the total IgE in the tissue and the total IgE in the serum. So we can say that there is a high concentration of IgE and frequency of IgE only within the polyp and not outside, which may mean that IgE is not coming from the blood, because otherwise we should see an increase in IgE and IgE positive cells also in the surrounding tissue. When we analyze the histology of these polyps, we could see that there are several cell agglomerates, and we analyze the contents of these agglomerates, and we could see that they are filled of B cells that are proliferating. They express AID, which is the aminase involved in hypermutation and class switch recombination. IgE is mainly observed in the surrounding tissue and not within the polyps. And in contrast, IgE was mainly observed, as I told you before, only within the polyps. Here you can see a co-staining of IgE and CD138 because we aim to analyze the production of IgE or the presence of IgE positive B cells or plasma cell within the polyps. And you can see here, we can see some plasma cell within the polyp. These are the double positive cells. These are the IgE positive CD138 negative cells with a beloved nucleus, which are eosinophils. And of course, again, in the adjacent tissue, we couldn't see any positive cells, either in the same patients or in some other controls, like in Crohn's disease, that it is another inflammatory condition related with the colon. And we also see that there is an accumulation. This is a germinal center, but the accumulation of these IgE positive cells is in the extrafollicular area, which may indicate that these IgE or these cells are generating within the germinal center. So for that, we analyze the content of these cell agglomerates, and for that, we carry out laser dissection microscopy. We obtain the stuff from these cellular agglomerates, and then we isolate RNA, and by RT-PCR and qPCR, we analyze the content in terms of the biology of the IgE in the germinal centers. And so we made a huge effort to find or trying to slice the polyp just where we have a germinal center, and to confirm that, we have to analyze AID expression, and we have a complete series of results in 3 patients in which we analyze the contents or the RNA in the whole polyp tissue in the germinal centers isolated, as I told you before, and also in the surrounding control tissue. So we selected these 3 polyps or these 3 patients because we found that there is a germinal center in the material, or the material is coming from a germinal center. To confirm from this patient, we could see that there are IgE positive cells in the tissue, only within the polyps and not in the surrounding control tissue. And to analyze the synthesis of IgE, we assessed different markers for transient class switch recombination, and so we used different primers, a set of primers to analyze germline transcript or sterile transcript, which is the signal when the class switch recombination initiates to locate the aminase, which then act and form and cleave the DNA to form the circle transcript. Both components are the sterile transcript and the circle transcript are transiently transcribed, and so our focus was to detect these 2 markers, and then we also analyzed to confirm that the class switch recombination has undergone a primer for the mature heavy change. So you know that there are 2 mechanisms to obtain the IgE. One is the direct class switch recombination from mu to epsilon, and then we have the sequential recombination class switch from mu to gamma and then gamma to epsilon. So to detect the direct class switch from mu to epsilon, we analyze at first the sterile transcript or the germline transcript, and we could see here that only in this patient the germline, the germinal center contained this, or the primer was positive for the RNA in the germinal center from this patient and not for the other. But when we analyze the circle transcript mu to epsilon, we could see, or we could evidence the presence of them in the germinal center. So we can see that there is a direct class switch recombination from mu to epsilon in this germinal center. And we didn't evidence any class switch recombination in the adjacent tissue. We did exactly the same from the sequential class switch recombination. This is gamma, mu to gamma, and so we could see that there is a class switch from mu to gamma 1, mu to gamma 3, and mu to gamma 4. And then we analyzed the same in terms of a circle transcript, gamma 1 to epsilon, gamma 3 to epsilon, and gamma 4 to epsilon, and we could detect the transcript in all of these samples, and so we could see, we could evidence that class switch, the recombination from gamma to epsilon, is also produced in these tissues. And finally, to confirm, we could analyze the different mature markers for the different isotypes, and we could see, and this is quite striking for us, because we could see that in any individual germinal center, we could evidence the production of class switch to generate IgM, IgD, IgE, IgG1. So in any individual germinal center, we could see that different isotypes are being produced, in some of them also IgA1 and IgA2, but in the adjacent tissue, we never observed the presence of IgE, but we could see the presence of the different isotypes. So in any individual germinal center, we could see that there is a simultaneous synthesis or production of the different isotypes, and mainly the IgE. So here we have the direct evidence that IgE is being produced within the polyps in the germinal center through these two mechanisms. So just to finish, the stroma of polyps contains a dense allergic cell infiltration with many eosinophils as the predominant cell components. Type 2 cytokines, CCL23, IL-33, and TSLP are upregulated only within the polyp. IgE is synthesized through direct and sequential class suite recombination mechanisms in the colonic tissue, which may mean that the polyp is working as a lymph node. So the question is, what is the function for the mesenteric lymph nodes in the colon? And this may have different impacts on treatments. So the mucosal therapeutic intervention could create a tolerogenic mucosal environment to modulate these type 2 inflammatory cells that are sustaining the allergic reaction. And the induction of a tolerogenic tissue environment could prevent or reverse the context for IgE, mainly to reverse this inflammatory context within the polyps. And so we are working also on different immunotherapeutic strategies in IgE-dependent mouse models. We have developed different protocols to induce Th1 activation, that is to immune modulate, or also to induce Tregs, and all of them are working to reverse this Th2 allergic inflammation that is observed in the intestinal tissue. So this is our working model in the food allergy context within the polyps. We have observed that there is a high content of type 2 cytokines. Different alarmins are produced. We could observe Th2 food-specific cells, B cells that are producing IgE, a high content of eosinophils and mast cells. And so now we have several questions why IgE is produced only within the polyps and not in the other parts of the colon, which are the cell sources for these type 2 cytokines. Are ILC2 involved? We are starting a project to analyze ILC2. What is the role for epithelial cells, which we think that this is the main cell triggering for all of this process? And mainly, are IgE memory B cells increase in human? We don't know exactly if these cells exist in human, if they are persistent in our tissue, and we don't know if this is the cause of the allergy in human and not in mice, because there is no IgE memory B cells in mouse. And so this is the approach to develop any tolerogenic immunotherapy, trying to reverse the inflammatory context within the polyp, and in that case, we think that we could restore the tolerance, and for that to reverse the clinic of these patients. So I want to thank the patients, the lab member. This work has been mainly done, as I told you before, by Karina Canciani, but also Cecilia Mulia, Renata Cuccialelo, Julian Baccaro, and Emanuel Feregotti are working now in this work to get the last results. And I also want to thank FOCIS, ALACI, and Argentinian Society for Immunology. And just a very brief and final announcement for the next meeting in Cuba, in Varadero, that will be held in June, not in February. It has been postponed from 6 to 10 of June, and what will be a great opportunity to resume the social, academic, and scientific interaction with all of you. Thank you very much.
Thank you very much, Guillermo, for a really nice talk, very, very interesting. So now I think you can start the discussion. And you have here 2 questions. One question is from Luisa Lemos to Ana. Is the fact of cross-fostering related just to the milk consumption or the microbiota from the foster mother?
Well, Luisa, that's a good question. We don't know. We are now trying. Actually, my student Carla was supposed to re-perform these experiments again, but with the pandemics, we could not do that. But we have to do the experiment because the two options could be true. The weaning itself and the establishment of the microbiota, and then it can get the microbiota from the foster mother, or there are components in the milk that are different from NOD and NOR mice, or the combination of the two. But we really don't know yet. I think my guess would be that both components would be very important, and the microbiota of these mice would be different because they are like housed with a different mouse, and there are differences in microbiota from NOR and NOD mice.
Okay. The next question is from Francisco Javier Turrubiates Hernandez. Are there any polymorphisms that induce lower expression of tight junction proteins?
You mean in the susceptible mouse, in NOD mice? Yeah. No, not that I know of. Actually, NOD mice had a lot of Iddm genes, but these are not one of them. And since those results are very new, I mean, nobody have looked at the intestinal mucosa before as something that would be relevant for diabetes induction. They look for other kinds of genes, but that's a good question, and it would be nice to look in the genome and see whether you have polymorphism in there, because it looks like it's something genetic. I agree, because we found it almost in a neonatal mouse. So it's one mouse that are 1 week old, they already have all these differences in the expression of these proteins. So it's a very nice idea. So we have to look, I don't know.
And it could be one of the causes for the translocation, no?
No, that's true.
And very early life.
Yeah, and it looks like it's something genetic. I think the question is very good. He's right. We should look in the genome of NOD mice and see whether we find something like that.
Okay, here we have Alderes. Thanks, Ana and Guillermo, for the brilliant talks. Question to Guillermo. It's very impressive, the association of eosinophils and type 2 cytokines with IgE. There have been studies by Boncourt's group associating antibody production and with gamma delta T cells as well. Can gamma delta T cells be found in the polyps? This is the first question. Do you have any evidence of gamma delta T cells in cytokine production and interplay with IgE production?
No, we have no idea. We didn't look for this delta cell so far.
Frozen.
And we have several cell targets to analyze, and I agree that it could be also relevant to analyze these cells. We lost Claudia, I think. Can I ask you a question, Anna?
Yeah, I also have a question for you. Okay. Go ahead. You ask first.
Did you see if there is also translocation to the liver?
We didn't look. That would be nice to do too. We looked at the pancreas because we wanted to see whether this bacteria would have a direct impact on the pancreas. And there is this route, right, of the lymphatic circulation, but we didn't look. Would be nice to look. That's another good idea for the next experiment.
And do you think it could be possible to restore tolerance in these mice by any other procedure?
Well, this what we don't know if it would be possible to completely restore tolerance. because it seems that there are defects that they have that are really, really early in life. But we saw that there are defects that appear later on. For example, the regulatory T cells, the defect in the generation of regulatory T cells seem to be later on. So it gives us the opportunity, as you said, to intervene at least in this. So maybe if we use a kind of, as you are using for your approach on allergic cells, if we use like probiotics or any kind of, or even like metabolites like butyrate or acetate or something anti-inflammatory, maybe we can reverse. Yeah.
Great.
Now, can I ask a question? First, this is fantastic to see this in a human. It's really absolutely fantastic. So congratulations, a beautiful work. And I was wondering, it's really striking to see these polyps and to imagine that they are really producing IgE and you have germinal centers there. And have you ever looked at the FH cells there, the follicular cells, the ones that you have in the germinal centers that would be fundamental for the class switch and all this? You didn't, right?
Not yet, not yet. We have recently obtained a grant to analyze the germinal center, and this is one of our aims. So I consider that these are preliminary data that we have, and now we have to move, but at least it's quite striking. But the main question for me is why only within the polyp and not in the area?
Yeah.
So I think that the epithelial cells, which are undoubtedly modified just to produce the polyp, they are mainly or heavily involved in this process. But so far, we don't have any other clue about which are the mechanisms that are behind to produce the cytokines and then to produce the IgE positive cells.
It looks like they are a kind of tertiary lymphoid organ, right? You
That's right.
create like a follicle. You call polyp, but it's really a follicle that is a proxy for a lymph node, for example.
Yes.
So that's what strikes me. And it would be nice to see whether they have really the structure like TF8 cells and also TFR cells, the regulatory cells, because there are work showing that Tregs can differentiate into TF-reg cells, so regulatory cells that regulate the production of, so would be nice to see how developed these follicles are in terms of germinal.
Yes, and to do this type of cytometry analysis, we will have to analyze the whole content of the, not only the germinal center. For that, we can only do is PCR or RT-PCR because it's impossible to isolate these structures.
Okay, thank you.
Sorry, my internet is really, really bad today. Sorry. So did you already answer no? The question of, well, I can imagine about gamma T cells. Okay, good. So I don't have any question more here, but I am curious about the talk of Guillermo about the polyps. If you have the chance to analyze another kind of polyps that are not by allergy, caused by allergy or something.
No, not yet. We will have to hear me. No. We didn't analyze any other type of tumoral polyp, at least, or from an adult people. But yeah, we know that we should do it. Yeah. But I don't think that we will find the same picture in any other tumoral polyp.
And it's very striking that they are in the column as well.
Only in the rectum, only in the rectum.
Yeah.
Not different sections of the large bowel.
Okay, I'll be look forward to read the rest of the story.
Me too.
it's already time to finish our seminar. Thank you very, very much for your presence here and for the really nice talk. Sorry for my internet that are really unstable today. So hopefully we can meet each other in the next month in some meeting here in Brazil, in Cuba, or whatever. So very, very nice, and thank you very much for your presence here.
Thank you very much for the invitation. It was really nice to meet you, at least in the internet.
Same. Thank you very much.
Okay, bye guys.
Bye-bye.
Thank you. Bye. E aí, Aninha, tudo bem?
Pronto, Gabs.
Val, você está aí? Rafaela?